Fatty acids. Among the simplest lipids are the fatty acids, which are used to synthesize triglycerides and phospholipids. Fatty acids can also be catabolized to generate adenosine triphosphate. A fatty acid consists of a carboxyl group in a hydrocarbon chain. Fatty acids can be either saturated or unsaturated. A saturated fatty acid contains only single covalent bonds between the carbon atom of the hydrocarbon chain. Because they lack double bonds, each carbon atom of the hydrocarbon chain is saturated with hydrogen atoms. For example, palmitic acid. An unsaturated fatty acid contains one or more double covalent bonds between the carbon atoms of the hydrocarbon chain. Thus, the fatty acid is not completely saturated with hydrogen atoms. For example, oleic acid. The unsaturated fatty acids have a kink or bend at the site of the double bond. If the fatty acid has just one double bond in the hydrocarbon chain, it is monounsaturated and it has just one kink. If a fatty acid has more than one double bond in the hydrocarbon chain, it is a polyunsaturated and it contains more than one kink. Triglycerides. The most plentiful lipid in your body and in your diet are the triglycerides, also known as triglycerols. A triglyceride consists of two types of building blocks, a single glycerol molecule and three fatty acid molecules. A 3-carbon glycerol molecule forms the backbone of a triglyceride. 3 fatty acids are attached by dehydration synthesis reactions, one to each carbon of the glycerol backbone. The chemical bond formed where each water molecule is removed is an ester linkage. The reverse reaction, hydrolysis, breaks down a single molecule of a triglyceride into 3 fatty acids and glycerol. Triglycerides can be either solids or liquids at room temperature. A fat is a triglyceride that is a solid at room temperature. A fatty acid of a fat are mostly saturated. Because these saturated fatty acids lack double bonds in their hydrocarbon chain, they can closely pack together and solidify at room temperature. A fat that mainly consists of saturated fatty acids is called a saturated fat. Although saturated fats occur mostly in meats, especially red meats and non-skim dairy products, whole milk, cheese, and butter, they're also found in a few plant products, such as cocoa butter, palm oil, and coconut oil. Diets that contain large amounts of saturated fats are associated with disorders such as heart disease and colorectal cancer. The oil is a triglyceride that is liquid at room temperature. An oil. An oil is a triglyceride that is a liquid at room temperature. The fatty acids of an oil are mostly unsaturated. Recall that unsaturated fatty acids contain one or more double bonds in their hydrocarbon chains. The kinks at the site of the double bonds prevent the unsaturated fatty acid of an oil from closely packing together and solidifying. The fatty acids of an oil can either be monosaturated, The kinks at the site of the double bonds prevent the unsaturated fatty acids of an oil from closely packing together and solidifying. The fatty acids of an oil can either be monounsaturated or polyunsaturated. Monounsaturated fats contain triglycerides that mostly consist of monounsaturated fatty acids. Olive oil, peanut oil, canola oil, most nuts and avocados are rich in triglycerides with monounsaturated fatty acids. Polyunsaturated fats contain triglycerides that mostly consist of polyunsaturated fatty acids. Corn oil, safflower oil, sunflower oil, soybean oil, and fatty fish, which is salmon, tuna, and mackerel, contain a high percentage of polyunsaturated fatty acids. Both monounsaturated and polyunsaturated fats are believed to decrease the risk of heart disease. Triglycerides are the body's most highly concentrated form of chemical energy. Triglycerides perform more than twice as much energy per gram as do carbohydrates and proteins. Our capacity to store triglycerides in adipose, fat tissue, is unlimited for all practical purposes. Excess dietary carbohydrates, proteins, fats, and oils all have the same fate. They are deposited in adipose tissues as triglycerides. The clinical connection: fatty acids and health and disease. As its name implies, a group of fatty acids called essential fatty acids, or EFAs, is essential to human health. However, they cannot be made by the human body and must be obtained from foods or supplements. Among the most important EFAs are omega-3 fatty acids, omega-6 fatty acids, and CIS fatty acids. Omega-3 and omega-6 fatty acids are polyunsaturated Omega-3 and omega-6 fatty acids are polyunsaturated fatty acids that are believed to work together to promote health. They may have a protective effect against heart disease and stroke by lowering total cholesterol, raising HDL, high-density lipoproteins, or good cholesterol, and lowering LDL, low-density lipoproteins, or bad cholesterol. In addition, omega-3 and omega-6 fatty acids decrease bone loss by increasing calcium utilization by the body, reduce symptoms of arthritis due to inflammation, promote wound healing, improve certain skin disorders, psoriasis, eczema, and acne, and improve mental function. Primary sources of omega-3 fatty acids include flaxseed, fatty fish oils that have large amounts of polyunsaturated fatty acids, fish oils, and walnuts. Primary sources of omega-6 fatty acids include most processed foods, cereals, breads, white rice, eggs, baked goods, oils with large amounts of polyunsaturated fatty acids, and meat, especially organ meat such as liver. Such an unsaturated fatty acid is called a CIS fatty acid. Note that the hydrogen atoms on either side of the double bond in oleic acid are on the same side of the unsaturated fatty acid. Such an unsaturated fatty acid is called a CIS fatty acid. CIS fatty acids are nutritionally beneficial unsaturated fatty acids that are used by the body to promote hormone-like regulators in cell membranes. However, when CIS fatty acids are heated, pressurized, and combined with a catalyst, usually nickel, in a process called hydrogenation, they are changed to unhealthy trans fatty acids. In trans fatty acids, the hydrogen atoms are on opposite sides of the double bond of an unsaturated fatty acid. Hydrogenation is used by manufacturers to make vegetable oils solid at room temperature and less likely to turn rancid. Hydrogenated or trans fatty acids are common in commercially baked goods: crackers, cakes, and cookies, salty snack foods, some margarines, and fried foods, donuts, and French fries. When oil is used for frying, and if the oil is reused, like in fast food French fry machines, CIS fatty acids are converted to trans fatty acids. If a product label contains the words hydrogenated or partially hydrogenated, then the product contains trans fatty acids. Among the adverse effects of trans fatty acids are an increase in total cholesterol, a decrease in HDL, increase in LDL, and an increase in triglycerides. These effects, which can increase the risk of heart disease and other cardiovascular diseases, are similar to those caused by saturated fats. On that note, it is said that and it's true, that hydrogenated fats and partially hydrogenated fats do not exist any place in nature. The only way that you can get them is by man, in the process described. So in that they don't exist anywhere in nature, you can't digest them. And so that's why they're particularly bad for you and cause the diseases mentioned. Our next chapter here is going to be phospholipids, followed by steroids. Other lipids, proteins, amino acids, polypeptides. I'm going to blast through this chapter. But right now, I'm going to take a break, and I will be back. Phospholipids. Like triglycerides, phospholipids have a glycerol backbone and two fatty acid chains attached to the first two carbons. In the third position, however, a phosphate group links a small charged group that usually contains nitrogen to the backbone. This portion of the molecule is polar and can form hydrogen bonds with water molecules. The two fatty acids, by contrast, are nonpolar and can interact only with other lipids. Molecules that have both polar and nonpolar parts are said to be amphipathic. Amphipathic phospholipids line up tail to tail in a double row to make up much of the membrane that surrounds each cell. In the synthesis of phospholipids, two fatty acids attach to the first two carbons of the glycerol backbone. A phosphate group links a small charged group to the third carbon in glycerol. In the circle represents the polar head, and the two wavy lines represent the two polar tails. Double bonds in fatty acid hydrocarbon chain often form kinks in the tail. That was just an illustration that I read, which is a repeat of the previous paragraph, and it explains how the polar head and the non-polar tail of each phospholipid combine to form membranes, which is very interesting. Steroids. The structure of steroids differs considerably from that of the triglycerides. Steroids have 4 rings of carbon atoms. Body cells synthesize other steroids from cholesterol, which has a large nonpolar region consisting of the 4 rings and a hydrocarbon tail. In the body, the commonly encountered steroids, such as cholesterol, estrogen, testosterone, cortisol, bile salts, and vitamin D, are known as sterols because they also have at least one hydroxyl alcohol group. The polar hydroxyl group makes sterols weakly amphipathic. Cholesterol is needed for cell membrane and structure. Estrogens and testosterone are required for regulating sexual function. Cortisol is necessary for maintaining normal blood sugar levels. Bile salts are needed for lipid digestion and absorption, and vitamin D is related to bone growth. In Chapter 10, we will discuss the use of anabolic steroids by athletes to increase muscular size, strength, and endurance. Now, it's really interesting that as I read, I'm going to have to backtrack it. In the body, the commonly encountered steroids such as cholesterol, estrogen, testosterone, cortisol, bile salts, and vitamin D are known as sterols because they also have at least one hydroxyl, one alcohol group. The polar hydroxyl groups make sterols weakly amphipathic. Cholesterol is needed for cell membrane structure. Cholesterol, which is synthesized in the liver, is the starting material for synthesis of other sterols in the body. Estradiol, cortisol, testosterone, cholesterol, and the hydrocarbon tail. Okay, so other lipids. Eicosanoids are lipids derived from a 20-carbon fatty acid called arachidonic acid. The two principal subclasses of eicosanoids are prostaglandins and leukotrienes. Prostaglandins have a wide variety of functions. They modify responses to hormones, contribute to the inflammatory process, prevent stomach ulcers, dilate, which is enlarge airways to the lungs, regulate body temperature, and influence formation of blood clots, to name just a few. Leukotrienes participate in allergic inflammatory responses. Other lipids are also fat-soluble vitamins such as beta-carotene, the yellow-orange pigments in egg yolk, carrots and tomatoes that are converted into vitamin A, vitamin D, E, and K, and lipoproteins. Proteins. Proteins are large molecules that contain carbon, hydrogen, oxygen, and nitrogen. Some proteins also contain sulfur. A normal, lean adult body is 12 to 18% protein. Much more complex in structure than carbohydrates or lipids, proteins have many roles in the body and are largely responsible for the structure of body tissues. Enzymes are proteins that speed up most biochemical reactions. Other proteins work as motors to drive muscular contraction. Antibodies are proteins that defend against invading microbes. Some hormones that regulate homeostasis also are proteins. Describe several important functions of proteins. Oh, the next table describes it. Excuse me. Which I will read. There are no illustrations, but the type of protein, structural proteins, and their function is to form structural framework of various parts of the body. Examples: collagen in bones and other connective tissues, and keratin in skin, hair, and fingernails. Regulatory function as hormones that regulate various physiological processes, control growth and development as neurotransmitters, mediate responses of the nerve system. So regulatory type of protein function as hormones that regulate various physiological processes, control growth and development as neurotransmitters, mediate responses of the nervous system. Examples: the hormone insulin, which regulates blood glucose levels, and a neurotransmitter known as substance P, which mediates cessation of pain in the nervous system. Contractile proteins allow shortening of muscle cells, which produces movement. Examples are myosin and actin. Immunological proteins. Immunological. Aid responses that protect body against foreign substances and invading pathogens. Examples are antibodies and interleukins. Transport proteins carry vital substances throughout the body, and an example of a transport protein is hemoglobin, which transports more oxygen and some carbon dioxide, not more oxygen. Transport proteins carry vital substances throughout the body, like hemoglobin, which transports most oxygen and some carbon dioxide in the blood. Catalytic protein act as enzymes that regulate biochemical reactions. Examples are salivary amylase, sucrase, and ATPase. Amino acids and polypeptides. The monomers of proteins are amino acids. Each of the 20 different amino acids has a hydrogen atom and 3 important functional groups attached to a central carbon atom, an amino group, an acidic carboxyl group, and a side chain, R group. At the normal pH of body fluids, both the amino group and the carboxyl group are ionized. The different side chain gives each amino acid its distinctive chemical identity. A protein is synthesized in stepwise fashion. One amino acid is joined to a second. A third is then added to the first two, and so on. The covalent bond joining each pair of amino acids is a peptide bond. It always forms between the carbon and carboxyl group of one amino acid and the nitrogen of the amino group of another. As the peptide bond is formed, a molecule of water is removed, making this a dehydration synthesis reaction. Breaking a peptide bond, as occurs during digestion of dietary proteins, is a hydrolysis reaction. When two amino acids combine, a dipeptide results. Adding another amino acid to a dipeptide produces a tripeptide. Further, additions of amino acids result in the formation of a chain-like peptide. 4 to 9 amino acids, or polypeptide, 10 to 2,000 or more amino acids. Small proteins may consist of a large polypeptide chain with as few as 50 amino acids. Large proteins have hundreds or thousands of amino acids and may consist of 2 or more polypeptide chains folded together. Because each variation in the number of sequences of amino acids can produce a different protein, a great variety of proteins is possible. The situation is similar to using an alphabet or 20 letters to form words. Each different amino acid is like a letter, and the various combinations give rise to a seemingly endless diversity of words. Peptides, polypeptides, and proteins. Levels of structural organization in proteins. Proteins exhibit 4 levels of structural organization. The primary structure is the unique sequence of amino acids that are linked by covalent peptide bonds to form a polypeptide chain. A protein's primary structure is genetically determined, and any changes in a person's amino acid sequence can have serious consequences for body cells. In sickle cell disease, for example, a nonpolar amino acid replaces a polar amino acid, glutamate, through 2 mutations in the oxygen-carrying protein hemoglobin. This change of amino acid diminishes hemoglobin's water solubility. As a result, the altered hemoglobin tends to form crystals inside red blood cells, producing deformed sickle cell shapes cells that cannot properly squeeze through narrow blood vessels. I'm going to read that last portion again. As a result, the altered hemoglobin tends to form crystals inside red blood cells, producing deformed sickle-shaped cells that cannot properly squeeze through narrow blood vessels. The symptoms and treatment of sickle cell disease are discussed on page 711. The secondary structure of a protein is the repeated twisting or folding of neighboring amino acids in the polypeptide chain. 2 common secondary structures are alpha helixes, clockwise spirals, and beta pleated sheets. The secondary structure of a protein is stabilized by a hydrogen bond, which form at regular intervals along the polypeptide backbone. The tertiary structure refers to the 3-dimensional shape of a polypeptide chain. Each protein has a unique tertiary structure that determines how it will function. The tertiary folding pattern may allow amino acids at opposite ends of the chain to be close neighbors. Several types of bonds can contribute to a protein's tertiary structure. The strongest but least common bonds, S covalent bonds called disulfide bridges, form between the sulfhydryl groups of 2 monomers of the amino acid cysteine. Many weak bonds, hydrogen bonds, ionic bonds, and hydrophobic interactions also help determine the folding pattern. Some parts of a polypeptide are attached to water, hydrophilic, and other parts are repelled by it, hydrophobic. Because most proteins in our body exist in watery surroundings, the folding process places most amino acids with hydrophobic side chains in the central core away from the protein surface. Often helper molecules known as chaperones aid the folding process. In those proteins that contain more than one polypeptide chain, not all of them do, the arrangement of the individual polypeptide chain relative to one another is the quaternary structure. I'll read that again. In those proteins that contain more than one polypeptide chain, not all of them do, the arrangement of the individual polypeptide chains relative to one another is the quaternary structure. The bond that holds polypeptide chains together are similar to those that maintain the tertiary structure. Proteins vary tremendously in structure. Different proteins have different architectures and different three-dimensional shapes. The variation in structure and shape is directly related to the diverse functions. In practically every case, the function of a protein depends on its ability to recognize and bind to some other molecule. Thus, a hormone bonds to a specific protein on a cell in order to alter its function, and an antibody protein binds to a foreign substance, an antigen, that has invaded the body. A protein's unique shape permits it to interact with other molecules to carry out a specific function. Homeostatic mechanisms maintain the temperature and chemical composition of body fluids, which allow body proteins to keep their proper three-dimensional shapes. If a protein encounters an altered environment, it may unravel and lose its characteristic shape. Secondary, tertiary, and quaternary structure. This process is called denaturation. Denatured proteins are no longer functional, although in some cases denaturation can be reversed. A frying egg is a common example of a permanent denaturation. In a raw egg, the soluble egg white protein albumin is a clear viscous fluid. When heat is applied to the egg, the protein denatures, becomes insoluble, and turns white. Enzymes. In living cells, most catalysts are protein molecules called enzymes. Some enzymes consist of two parts. A protein portion called an apoenzyme and a non-protein portion called a cofactor. The cofactor may be a metal ion such as iron, magnesium, zinc, or calcium, or an organic molecule called coenzyme. Coenzymes often are derived from vitamins. The names of enzymes usually end in the suffix "ase." All enzymes can be grouped according to the types of chemical reactions they catalyze. For example, oxidase, add oxygen; kinase, add phosphate; dehydrogenase, remove hydrogen; ATPase, split ATP; anhydrase, remove water; protease, break down protein; and lipase, break down triglycerides. Enzymes catalyze specific reactions. They do so with great efficiency and with many built-in controls. Three important properties of enzymes are as follows. Enzymes are highly specific. Each particular enzyme binds only to specific substrates, the reactant molecule on which the enzyme acts. Of the more than 1,000 known enzymes in your body, each has a characteristic three-dimensional shape with a specific surface configuration, which allows it to recognize and bind to certain substrates. In some cases, the part of the enzyme that catalyzes the reaction, called the active site, is thought to fit the substrate like a key fits into a lock. In other cases, the active site changes its shape to fit snugly around the substrate once a substrate enters the active site. This change in shape is known as an induced fit. Not only is an enzyme matched to a particular substrate, it also catalyzes a specific reaction. From among the large number of diverse molecules in a cell, an enzyme must recognize the correct substrate and then take it apart or merge it with another substrate to form one of the more specific products. Enzymes are very efficient. Under optimal conditions, enzymes can catalyze reactions at rates that are from 100 million to 10 billion times more rapid than those of similar reactions occurring without enzymes. The number of substrate molecules that a single enzyme molecule can convert to produce molecules in 1 second. The number of substrate molecules that a single enzyme molecule can convert to product molecules in 1 second is generally between 1 and 10,000 and can be as high as 600,000. Enzymes are subject to a variety of cellular controls. The rate of synthesis and their concentration at any given time are under the control of a cell's genes. Substances within the cell may either enhance or inhibit the activity of a given enzyme. Many enzymes have both active and inactive forms in cells. The rate at which the inactive form becomes active or vice versa is determined by the chemical environment inside the cell. Enzymes lower the activation energy of a chemical reaction by decreasing the randomness of the collisions between molecules. They also help bring the substrates together in the proper orientation so that the reaction can occur. The substrate makes contact with the active site on the surface of the enzyme molecule, forming a temporary intermediate compound called the enzyme-substrate complex. In this reaction, the two substrate molecules are sucrose, a disaccharide, and water. The substrate molecule are transformed by the rearrangement of existing atoms, the breakdown of the substrate molecule, or the combination of several substrate molecules into the product of the reaction. Here the products are two monosaccharides, glucose and fructose. After the reaction is complete and the reaction products move away from the enzyme, the unchanged enzyme is free to attach to other substrate molecules. Sometimes a single enzyme may catalyze a reversible reaction in either direction, depending on the relative amount of the substrate and products. For example, the enzyme carbonic anhydrase catalyzes the following reversible reaction. Carbon dioxide plus water equals carbonic acid. Or carbonic acid yields water and carbon dioxide. During exercise, when more CO2 is produced and released into the blood, the reaction flows to the right, increasing the amount of carbonic acid in the blood. Then as you exhale CO2, its levels in the blood falls and the reaction flows to the left, converting carbonic acid to CO2 plus water. Our next chapter is nucleic acid, deoxyribose nucleic acid, DNA, and ribonucleic acid. Nucleic acid, so named because they were first discovered in the nuclei of cells, are huge organic molecules that contain carbon, hydrogen, oxygen, nitrogen, and phosphorus. Nucleic acids are of two varieties. The first, deoxyribose nucleic acid, forms the inherited genetic material inside each human cell. In humans, each gene is a segment of DNA molecule. One gene determines the traits we inherit, and by controlling protein synthesis, they regulate most of the activities that take place in the body's cells throughout our lives. When a cell divides, its hereditary information passes on to the next generation of cells. Ribonucleic acid, the second type of nucleic acid, relays instructions from the genes to guide each cell's synthesis of proteins from amino acids. A nucleic acid is a chain of repeating monomers called nucleotides. Each nucleotide of DNA consists of 3 parts. A nitrogenous base, DNA contains 4 different nitrogenous bases which contain atoms of carbon, hydrogen, oxygen, and nitrogen. In DNA, the 4 nitrogenous bases are adenine, thymine, cytosine, and guanine. Adenine and guanine are larger double-ring bases called purines. Thymine and cytosine are smaller single-ring bases called pyrimidines. The nucleotides are named according to the base that is present. For instance, a nucleotide containing thymine is called a thymine nucleotide. One containing adenine is called an adenine nucleotide, and so on. Pentose sugar, a 5-carbon sugar called deoxyribose, attaches to each base in DNA. Phosphate group. Phosphate groups alternate with pentose sugars to form the backbone of a DNA strand. The bases project inward from the backbone chain. 1953, F.H.C. Crick of Great Britain and J.D. Watson, a young American scientist, published a brief paper describing how these 3 components might be arranged in DNA. Their insights into data gathered by others led to construct a model so elegant and simple that the scientific world immediately knew it was correct. In the Watson-Crick double helix model, DNA resembles a spiral ladder. 2 strands of alternating phosphate groups in deoxyribose sugar form the upright of the ladder. Paired bases held together by hydrogen bonds form the rungs. Because adenine always pairs with thymine and cytosine always pairs with guanine, if you know the sequence of base in one strand of DNA, you can predict the sequence of the complementary second strand. Each time DNA is copied, as with living cells dividing to increase their number, the 2 strands unwind. Each strand serves as the template or mold on which to construct a new second strand. Any change that occurs in the base sequence of a DNA strand is called a mutation. Some mutations can result in the death of a cell, cause cancer, or produce genetic defects in future generations. RNA, the second variety of nucleic acid, differs from DNA in several respects. In humans, RNA is single-stranded. The sugar in RNA nucleotide is the pentose ribose. And RNA contains the pyrimidine base uracil instead of thiamine. Cells contain 3 different kinds of RNA: messenger RNA, ribosomal RNA, and transfer RNA. Each has a specific role to perform in carrying out the instructions coded in DNA for protein synthesis. I'm going to read that again. RNA, the second variety of nucleic acid, differs from DNA in several respects. In humans, RNA is single-stranded. The sugar in the RNA nucleotide is the pentose ribose, and RNA contains the pyrimidine base uracil instead of thiamine. Cells contain 3 different kinds of RNA: messenger RNA, ribosomal RNA, and transfer RNA. Each has a specific role to perform in carrying out the instructions coded in DNA for protein synthesis. Okay. Now, the clinical connection, DNA fingerprinting, is an interesting little article here. A technique called DNA fingerprinting is used to research and in a court of law to ascertain whether a person's DNA matches the DNA obtained from samples or pieces of legal evidence such as blood stains or hairs. In each person, certain DNA segments contain base sequences that are repeated several times. Both the number of repeat copies in one region and the number of regions subject to repeat are different from one person to another. DNA fingerprinting can be done with minute quantities of DNA. for example from a single strand of hair, drop of semen, or a spot of blood. It also can be used to identify a crime victim or a child's biological parent, and even to determine whether two people have a common ancestor. Okay, I'm going to move on to adenosine triphosphate. Adenosine triphosphate, ATP, is the energy currency of the living systems. ATP transfers the energy liberated in exergonic. That word's not working out for me. Tried it several times. It's just not working out. Exergonic. Now I have to start over because I don't want to confuse myself when I listen to this later. Adenosine triphosphate is the energy currency of living systems. ATP transfers the energy liberated in exergonic catabolic reactions to power cellular activities that require energy. Exergonic reactions. Among these cellular activities are muscular contractions, movement of chromosomes during cell division, movement of structures within cells, transport of substances across cell membranes, and synthesis of large molecules from smaller ones. As its name implies, ATP consists of 3 phosphate groups attached to adenosine, a unit composed of adenine and the 5-carbon sugar ribose. When a water molecule is added to ATP, the 3rd phosphate group, symbolized by P in the following discussion, is removed and the overall reaction liberates energy. The enzyme that catalyzes the hydrolysis of ATP is called ATPase. Removal of the 3rd phosphate group produces a molecule called adenosine diphosphate in the following reaction. ATP, adenosine triphosphate plus water, and ATPase yields adenosine diphosphate plus phosphate group plus energy. As noted previously, the energy supplied by the catabolism of ATP into ADP is constantly being used by the cell. As the supply of ATP at any given time is limited, a mechanism exists to replenish it. The enzyme ATP synthase catalyzes the addition of a phosphate group to ADP in the following reaction. Adenosine diphosphate plus phosphate group plus energy in the presence of ATP synthase yields adenosine triphosphate plus water. Where does the cell get the energy required to produce ATP? The energy needed to attach a phosphate group to ADP is supplied mainly by catabolism of glucose in a process called cellular respiration. Cellular respiration has 2 phases, anaerobic and aerobic. Anaerobic phase is a series of reactions that do not require oxygen. Glucose is partially broken down by a series of catabolic reactions into pyruvic acid. Each glucose molecule that is converted into a pyruvic acid molecule yields 2 molecules of ATP. Aerobic phase. In the presence of oxygen, glucose is completely broken down into carbon dioxide and water. These reactions generate heat and 36 or 38 ATP molecules. Chapter 10 and 25 cover the details of cellular respiration. In Chapter 1, you learn that the human body is comprised of various levels of organization. This chapter has just showed you the alphabet of atoms and molecules that is the basis for the language of the body. Now that you have an understanding of the chemistry of the human body, you're ready to form words. In Chapter 3, you will see how atoms and molecules are organized to form structures of cells and perform the activity of cells that contribute to homeostasis. And that would conclude Chapter 2, which ends in a test. And then we move on to the cellular level of organization, which will be Chapter 3. Good. Okay. So, that concludes this episode of Let's Read Anatomy and Physiology. And when I come back again, I will read Chapter 3, the cellular level of organization. Thank you for joining me. I hope you're getting something out of this. I hope you can pick up some tidbits of knowledge here and move on to bigger and better things.